Welcome to the second episode of Diabetes Bio. This is a new podcast for the Diabetes Journal, which is the flagship research publication of the American Diabetes Association. Each month, we will interview one or more authors of the feature articles published in that month's issue of the Diabetes Journal. We will also have a Sweet Talk segment of the podcast where we will discuss additional articles, conferences, major events in the field, or sometimes, like we did last month, we'll have a rapid fire exchange between the co-hosts to highlight other articles of interest in the journal. My name is Darleen Sandoval, and I'm joined by my podcast co-hosts, Dr. Kirk Haberger and Dr. Kevin Williams. How are you guys doing today?
Hey, we're doing good.
Yeah, doing really well.
I think we should all celebrate that we haven't been kicked off the air yet. They're still inviting us back, so this is a success.
Woohoo!
Number 2.
Kirk, why don't you tell us about the articles we'll be reviewing this month?
Sure, Darlene. Today we have 2 great original articles. We'll be hearing from Dr. Satya Dash and his work evaluating the role of the low-calorie sweetener erythritol on cardiometabolic disease. We also have the chance to sit down with Drs. Alan Attie and Jeffrey Hodgin to discuss their collaboration, which identified a potential genetic mechanism underlying obesity-associated diabetic nephropathy in the BTBR mouse.
Awesome. And Kevin, you're going to be hosting our Sweet Talk segment this month. Tell us about it.
Absolutely. Today, we're highlighting a brand new set of feature articles in the journal called Classics in Diabetes. This feature brings timeless articles from the past into the spotlight, and they're showcasing their value for today's scientists. In this episode, we're discussing the first Classics in Diabetes article published in September 2023 issue of the journal by Dr. Jonathan Campbell and Dr. Frans Schuit. This Classics in Diabetes article is actually based on work published by Dr. Schuit in the journal back in 1998 on rethinking the role of glucagon, pro-glucagon hormones, and insulin. And we have the pleasure today of discussing this topic with both Dr. Campbell and Dr. Schuit.
You know, as someone who focuses a lot of their work on glucagon and pro-glucagon hormones, I'm really excited about this one.
Yeah, definitely. Our first feature article from the February 2024 issue of Diabetes is titled Erythritol is a Potential Causal Contributor to Cardiometabolic Disease, a Mendelian Randomization Study. We are joined by the senior author of this paper, Dr. Satya Dash. Dr. Dash is senior scientist at Toronto General Hospital Research Institute and medical director of the bariatric surgery program at the University Health Network. Welcome, Dr. Dash. Congratulations on being selected as one of our feature papers of the month, and thank you so much for being a part of our podcast.
Thank you so much for the invitation.
So, Dr. Dash, we like to start with sort of an off-topic icebreaker, right? Just to get this going. Maybe you'd like to share with us a fun fact about yourself that has nothing to do with science.
Okay. I'm trying to learn Japanese, and it's been painful.
Very cool.
But I'm making slow progress, but I'm enjoying it.
That's really cool. Any particular reason for that? Just a language you wanted to learn, or?
It's a place I visit quite often. I try and go at least once a year, and I just love the culture and the heritage. And so the more I learn, the better my interactions with people there.
That's really cool. All right, so now on to the science discussion. Can you briefly summarize your research program and then talk about how this work has inspired you to investigate the potential relationship between erythritol and cardiometabolic disease?
Sure. My big sort of research goal, interest, is understanding why people develop obesity and why certain people develop cardiometabolic diseases. And in order to answer that question, I use a variety of approaches, population studies, including genetic studies. I also do some in vivo physiologic studies. And another sort of specific interest that I have is how do different people respond to bariatric surgery, which is one of my clinical interests, because there's so much variability with regards to type 2 diabetes remission, degree of weight loss. So that's a sort of broad overview of the sort of research that I'm interested in. And this is actually slightly different to my usual line of research, the erythritol story. I mean, as a clinician, I see a lot of people with obesity, a lot of people with type 2 diabetes, and they often use low-calorie sweeteners, artificial sweeteners, to manage their glycemia and their weight. There's a lot of press about sugar-sweetened beverages and how they contribute to obesity. And so I do try and keep up with the literature. And a big paper came out in Nature Medicine recently, which showed that across 3 cohorts, increased erythritol appeared to be independently associated with major adverse cardiovascular events. And so when I looked at the data, it was a very interesting paper, but there was some potential confounded with the observational data. But when they did the in vitro and ex vivo studies, there seemed to be very convincing evidence, at least in that setting, that erythritol was affecting platelet aggregation. And then when you look back to the sort of previous studies, there's a history, lots of studies showing that increased erythritol is associated with metabolic diseases such as type 2 diabetes. So I was interested from that perspective. But then the other thing that happened is this paper got a lot of press, which then patients come across. So a number of patients actually did come to me and ask me, is erythritol safe? It's an ingredient in stevia, which is a very commonly used low-calorie sweetener. And so that prompted me to look into this in a little bit more detail.
Very good, very good. Maybe it would be helpful, definitely for me, but for our audience, if you could expand on what exactly erythritol is, how it's used as a food additive. I know you mentioned stevia, but maybe just a little more background there would be helpful.
I also want to know if it's in my Diet Coke that I'm drinking right now.
So erythritol, it can come from endogenous sources or exogenous sources. So the endogenous sources it can come from, it is actually, you can get it from glucose metabolism through the pentose phosphate pathway. So erythritol is a 4-carbon polyol, and so it can be synthesized from there. There's some evidence that with oxidative stress, cells can produce erythritol through this pathway as well. But you can also get it exogenously. It's found in various fruits, vegetables, fermented foods, etc., but usually in low quantities. But because of its taste, especially in the last sort of 10 to 15 years, they're increasingly used in the food industry as a low-calorie sweetener because there's lots of awareness about the potential role of sugar-sweetened beverages in adiposity. So it's being used more and more in that setting.
Very good. Thanks.
So let's get a little bit deeper into the paper. One of the things that seemed to be a strength of this paper is the statistics that you used. So can you talk a little bit about the summary statistics that you used from the GWASs, and then maybe a little bit about the subject characteristics from those previous studies that you're getting the data from?
So perhaps I could start off with what Mendelian randomization sort of does. What you can do is if you have genetic variants which are very strongly associated with an exposure, so for example, in this case, erythritol. So we know what effect these particular variants have on erythritol concentration in European ancestries. All of these cohorts were of European ancestry. So we know that it's associated with erythritol. What we can then do is in other genome-wide association studies, which have been done, looking at various outcomes, such as type 2 diabetes, waist-hip ratio, BMI, coronary artery disease, we can look at the effect of these particular variants, which are associated with erythritol, to see what effects they have on these outcomes in these other studies. And so for each of these variants now, you can look at the effect that they have on the outcome of interest. So for example, type 2 diabetes, and you know what effect they're having on erythritol. And so you can then infer what effect an increase in erythritol may have on the outcome of interest. Now, the limitation with this approach, of course, is it has to be done in ancestries which are similar in both the exposure and the outcome. So we only use summary statistics from GWASs that had European ancestry because those are the most commonly used.
Very good. So I'd actually like to ask a sort of a follow-up there. And you mentioned that you used cohorts of European ancestry. Moving forward, do you expect similar results in cohorts that are of non-European ancestry? And is that something you plan to do?
That would be an important thing to do. And I think this is something that that is true for most genetic studies. Most of them have been done in European populations, but as time goes on, we're getting more and more non-European ancestries. The sample sizes are getting better. There are more metabolomic studies. And so when those studies become available, I think it would be important to repeat these analyses to make sure that they hold true in those populations as well.
Are there any other key findings that you'd like to discuss from your paper that you haven't already mentioned?
Absolutely. So there are with any data, there are some caveats and potential limitations, and I think it's worth exploring them so that will help us plan potentially future studies. Now, the first thing with all of the studies that have been published on erythritol, including the recent paper that looked at cardiovascular outcomes with erythritol and all of the prior studies, we don't actually have detailed information on whether the participants actually used exogenous erythritol, and if so, how much did they use? We don't have that information, so that's a limitation. But based on timelines, we can potentially infer that the GWASs were likely done in people who are not consuming large amounts of exogenous erythritol, just based on the timelines. The other thing that increases the probability that this is true is if you look at the nearest genes, the genes nearest to the SNPs that were significantly associated with erythritol. The nearest genes were things like transketolase, aldoketoreductase 1 family member 1, and these are genes that are likely implicated in the pentose phosphate pathway. So biologically, they're plausible regulators of endogenous erythritol. I suspect that our results apply to endogenous erythritol, and the previous studies have indicated that there is a linear relationship between erythritol and cardiometabolic outcomes. However, if you take exogenous erythritol, you can actually potentially get your erythritol levels up to 1,000-fold higher. And to my knowledge, no one has actually formally assessed whether there are nonlinear relationships between erythritol and cardiometabolic outcomes. Certainly in the recent Nature Medicine paper by Dr. Hazensbrough, the highest erythritol quartile seemed to have the highest cardiovascular risk, but they also had higher prevalence of baseline cardiovascular disease. So that could potentially be a confounder. So I think this is something that we obviously need to wait to see what the data shows. Is there a non-linear relationship? The other thing to mention as well is we used summary statistics from 3 cohorts, and there was a good reason for using the third one. And that is that erythritol and threotol are isomers of each other. And standard mass spectrometry can't differentiate between them. So if you're doing a metabolomic study, it's important to separate them first. And the largest GWASs with European ancestry that were done, they did use liquid chromatography with a particular type of column that would likely separate them. But they only did GWASs of erythritol. The third sort of study that we did, which was the Twins UK CORA study, they actually did a GWAS of erythritol and threotol. And the variants associated with erythritol did not associate with threotol. So that increases our confidence in the results. So I think the data suggests that endogenous erythritol is likely not a causal contributor, but I think we need a better understanding of the dose relationship between exogenous and cardiometabolic outcomes. And I think we probably need prospective studies, short to medium-term studies, to look at what the effects are. Because there have been some short-term studies done with erythritol, and there are potentially some beneficial effects. So for example, acutely, there's no increase in glucose if you take erythritol, but GLP-1 goes up. In people with type 2 diabetes, over a 4-week period, if you take it, endothelial function actually improves. So potentially beneficial effects, but then in ex vivo and in vitro, potentially deleterious effects on platelet aggregation. So we probably need prospective, well-powered longitudinal studies to look at this in a little bit more detail.
Great, thanks. So, Dr. Dash, have you changed how you consume erythritol or erythritol-containing foods based on your research, or are you still, let's say, agnostic to it?
Yeah, so I don't use Coke Zero, Dr. Sandoval. I don't use Diet Coke, Dr. Sandoval. I use Coke Zero. And I do sort of consume it from time to time if I do use it. And I do use aspartame-based artificial sweeteners. Now, there is some data suggesting that those are harmful as well, but in meta-analyses with the randomized controlled trials, so far I haven't seen any data that makes me concerned, but we wait for more data.
Well, Dr. Dash, thank you very much for sharing your expertise and your time with us today, and especially talking about your recent work and highlighting the role, or I guess lack thereof, of erythritol in cardiometabolic disease. I encourage the listeners to read the full article, as our featured articles are freely accessible online and are in the February issue of Diabetes. One of the feature articles from the February 2024 issue of Diabetes is titled Genetic Analysis of Obesity-Induced Diabetic Nephropathy in the BTBR Mouse. We're joined by two of the authors of this paper, Dr. Jeffrey Hodgin, Associate Professor at the University of Michigan, and Dr. Alan Attie, who is the Jack Gorski Professor of Biochemistry from the University of Wisconsin. Welcome, Drs. Hodgin and Attie. Congratulations on being selected, and thank you so much for being part of this podcast.
Thank you.
Thank you. It's a pleasure to be with you.
So, as a IU alum, I just want to start by saying it's nice that football season is finally over and that we can finally see Big Ten schools getting along, right? So I think this is a great example.
Well, I'm going to be an instigator and say, go blue.
Yay.
Thank you guys so much for being here. I want to start off by asking you more of a fun question, and you'll each have your chance. But if you could go back in time and choose a different career path that was unrelated to science, what would it be? We'll start with you, Alan.
It would be either psychology or photography.
Oh, interesting. Are those photos we can see in back of you some of your work?
Yes.
Nice.
Yeah,
Very
this is a black and white panorama of Death Valley.
Oh, that's awesome. And Jeff, what about you?
Can't think of anything outside of science. I can think of some other areas in science that as time has gone by, I'd be like, boy, that would have been nice. Like
Okay.
some of the more engineering focused areas. I particularly like what I see with restoring limb use in patients who have lost a limb or lost the use of it. That just seems like it could be so interesting and so rewarding. Although I think that the stuff I'm doing now is interesting and rewarding, but
All right. Thank you both for indulging us on that. So now we'll talk science. So, a question for both of you. Can you briefly summarize your research interests and then how those interests led to this collaboration? So, this time we'll start with Jeff.
okay, so I'm a kidney pathologist at the University of Michigan, but I've been working with the molecular aspect of kidney disease for some time, largely transcriptomics. I'm very interested in integrating the structural and the molecular aspects of the analysis for kidney disease, largely glomerular disease, but I'm very much involved with some large biopsy cohorts, human biopsy cohorts, such as the Kidney Precision Medicine Project, Nephrotic Syndrome Study Network called Neptune, the Accelerated Medicines Partnership for Lupus Nephritis, and also H3 Africa. And we're getting like tons of material to do structural and molecular analysis. And of course, correlating that with clinical features and clinical outcome. But this collaboration with Alan was actually the beginning of really doing structural analysis, in this case, obviously a mouse model, which I have a lot of experience with mouse models back in the day, and getting some, very detailed morphometric analysis of the glomerulus and in a very large data set. That was particularly exciting to be able to do that. Now, the genetic side is way out of my wheelhouse, which is, of course, why we have to have a collaboration. But it's been, incredibly interesting to see the data that's come out of this and that things that we can build on.
Yeah, that's great. Alan.
So I began my career in lipid metabolism. About halfway in, I decided I wanted to do something new and different. And I had been teaching metabolism for many years, and I love teaching, and I love teaching metabolism. And that made me follow the diabetes field. And I just felt like I'd been following it a long time, but not being in it, and I wanted to be in it. And so I thought, well, how in the world am I going to get into this field when there are already so many great people in it? They don't need me. What's my niche going to be? And so at that time, there were not very many people in mouse genetics studying diabetes, but I didn't really know anything about genetics either. So I went to the Jackson lab and I took their mouse genetics course, and I went to Cold Spring Harbor and took a bunch of courses. And then I took a plunge and decided I would use mouse genetics. And what we did was we landed on, and this was serendipitous, a mouse strain that when made obese with the leptin OB mutation was very, very diabetic. Whereas the black 6 mouse, if you don't stress it, is not diabetic when it's OB. And if you see hyperglycemia in those mice, like there are a lot of published papers that show a lot of hyperglycemia, it's because they're stressed. They have a lot of cortisol. But that's not really diabetes. That's just a lot of cortisol. that became our model system, and we used that to ask, what's the difference between obese people who are diabetic and obese people who are not diabetic? Because that's obesity diabetes dichotomy. Most people who have type 2 diabetes are obese, but most people who are obese don't have diabetes. And so we thought that we reproduced this dichotomy in mice, and we could use that to understand what the genetic basis was for that. And I just happened to be giving a seminar at the University of Washington some time ago, and Charlie Alpers raised his hand and said, "Have you ever looked at the kidneys?" And I said, "No." And he said, "Well, there's no good mouse model for diabetic nephropathy. Will you send me the kidneys so I can have a look?" And so I did. And he was breathless when he called me back a few days later. He was so excited because he had never seen the features that he found, which I'll let Jeff describe, in those kidneys that were the best he's ever seen in an animal model for human diabetic nephropathy.
Great. So, Alan, that was a wonderful introduction to my question. And that is, you're speaking of the leptin deficient BTBR mouse, correct?
That's correct.
And, maybe you can give us a little bit more historical overview and how this specific model has sort of enhanced our original interpretations of metabolism and diet-induced, or I guess just obesity, genetic obesity, right?
Well, I don't want to be too grandiose about that because leptin deficiency is not the same as regular obesity. Most people who are obese are not leptin deficient. In fact, it's the opposite. They have hyperleptinemia. So they're not equivalent, but I think of it more as a sensitizer. People ask me the question, if diabetes is so genetic and has high heritability, then why are we seeing a diabetes epidemic? Did the human population mutate? That is because non-scientists and even scientists don't fully understand the meaning of sensitizers. And so it is true that people who have type 2 diabetes have genetically crummy beta cells. But the reason that obesity matters is that, in fact, they had crummy beta cells 75 years ago before the obesity epidemic, but it didn't matter because they weren't asking so much from those beta cells. So the obesity epidemic is the real reason why we have a diabetes epidemic. We are asking the beta cells to do more. And so now the genetic effects produce a bottleneck. So basically, you have a sensitizer, which is obesity, creating the bottleneck, or actually another way to put it is changing the penetrance of the alleles that are contributing to diabetes.
Great. Thanks.
Okay, Jeff. So we've just heard a little bit about this mouse model and its replication of obesity and diabetes. Can you tell us a little bit about the nephropathy side of this mouse model and how it translates to the human pathology?
So, as you well know, models of diseases are crucial for preclinical study to help understand the disease and come up with new targets. And for decades, we've had not very good models for diabetic nephropathy in mice, largely because they just are so resistant to the progression of the disease for multiple reasons. It's specifically the B6 mouse, the black 6, is just so resistant to kidney stuff. It was a model that I used to use long ago for atherosclerosis. That worked, but not for kidney disease. And so you have to go to other strains like the BTBR. Now, in this case, we've had other strains of mice and like inject them with streptozotocin to make them diabetic and et cetera. But generally, we would see early changes that were applicable to the human disease, that being like expansion of the mesangium of the glomerulus, the mesangium of the glomerulus, or thickening of the glomerular basement membrane. But it was always very early in appearance. But with this mouse model, we not only see the early, but we also see later features, much greater thickening of the GBM, mesangiosclerosis that can appear nodular, these sort of Kimmelstein-Wilson nodules that you can see. And in addition, we see some, but not a great deal, but some of like changes in the vasculature and some tubular interstitial changes. That's still a problem with the models in that, diabetic nephropathy is not just glomerular, it's also tubular interstitial. And we don't see a lot of that in the mouse models unless we do a lot more to them, like make them hypertensive, et cetera. And even then it's challenging. But this one definitely shows early and later stages in the glomerular pathology.
How long does it take for this pathology to develop?
Right. So this model, in about 6 to 8 weeks, you'll start to see some changes in the glomerulus, and then you can go further from that. And in this study, 24 weeks, the time when they were euthanized at the later stage, which is well within a time frame that you can do. It's more feasible to do your experiments as opposed to a model that might take a year.
Yeah, definitely. Kirk?
So now I'd like to talk about the genetics, right? So I guess this goes back to Alan. Can you tell us about the genetic analysis that you guys used and how you arrived at the podocyte-specific gene nephrin-1?
Yes. So what we did, which we no longer do in our genetics anymore for reasons that I'll tell you in a moment, is what's called an F2 intercross. So you take the two strains and you breed them to make F1s, and then you breed the F1s and you get F2s. That allows you to have two episodes of meiosis that are discernible. You always have meiosis, but in this case, they are discernible because we have markers spanning the genome that are polymorphic. And so they tell you whether or not a portion of the genome came from grandma or grandpa. And then what we do is you generate a lot of animals. I think we had 400 or so in this particular case, and you score them. And then you look marker by marker to see whether or not the phenotype that Jeff scored and quantitated very painstakingly over a long period of time was associated with any particular markers. And when that happens, you compute a statistical score and you graph it. It's called a LOD score, which is called the log of the odds ratio. It's very basically like the negative log of the p-value. And then we have a correction for multiple testing. And so we look for anything above that. And we had only a single strong peak. We had some minor weak peaks, but we only had a strong peak. We probably would have gotten more if we had a larger sample size. Now, the limitation on an F2 intercross is very poor mapping resolution. And that's because it's not for two reasons. One is that with only two meioses, you have large blocks of the genome that are inherited as a big block. So no matter how many markers you have, the markers within any block are correlated, so they don't give you any more information. And then the other problem is that these mouse strains are all cousins anyway. So they have large parts of the genome that are identical by descent. And so you're not interrogating those parts of the genome. So we've corrected that in our current work where we've adopted a different model, which is an outbred stock derived from 8 mouse strains. So we have way more genetic diversity and they've been bred for 40 generations. So we have a lot more meioses and so way better mapping resolution. And instead of 4,000 markers, we have 150,000 markers. So we're supercharged now.
Yeah, I guess. So nephrin-1 had previously been identified as a kidney marker for pathology. Is that correct?
Yeah, so actually it's a very interesting story. So nephrin was the first gene that was found, a mutation leading to glomerular disease, being from the podocyte. At the time, this was actually a bit before my time, the podocyte didn't get a lot of attention. A lot of the research, I believe, was on the mesangium, et cetera. And then Carl Tryggvason found this mutation in nephrin causing nephrotic syndrome, childhood nephrotic syndrome. So then a lot of eyes turned to the podocyte. And since then, many, many genes have been discovered that have mutations causing dysfunction of the podocyte leading to kidney disease. And so finding nephrin in the area, the QTL for this study was a bit of find a little gold mine there to go, oh, that one, to really point to, the podocyte and genetics of the podocyte, because it's not expressed in any other cells in the kidney. So the podocyte is important here.
But I think it's important to point out that we are offering it as a candidate gene, that we did not prove that was the gene. And so that still remains. And I'm hoping Jeff will do the follow-up experiments to establish that. I don't know if Jeff is doing that or not. Are you doing that, Jeff?
Well, I was going to ask you about those kidneys from the mix of the 8 mouse strains you've mixed.
Yeah, let's talk about that later.
Real quick, Alan, I want to circle back to the use of the OBOB BTBR mouse.
Yes.
And what's the relationship between leptin and nephrin 1? Is there any, or was it just that you've used the leptin deficiency to push the mouse towards this nephropathy?
Well, it's the latter. However, a very important outcome of finding this model was some very beautiful work done by Charlie Alpers, and I think some other people too, showing that diabetic nephropathy is reversible. And he's done it several different ways. And one of them is by giving leptin back to the BTBR mice. And he's able to reverse the nephropathy with leptin.
That's
Very
really
cool.
interesting. Yeah.
And that's really amazing. I don't know, Jeff, was that the first demonstration that nephropathy could be reversible?
I believe so. Yeah.
So that was a landmark
In the mice.
study, and that's entirely Charlie. Just so the credit goes to the right place.
Yeah.
One of the things that really struck me when I started reading your paper was a statement about how, and I really should have known this, but a statement about how the severity of diabetes complications isn't necessarily driven by the severity of diabetes, right? And so that led to the justification that there could be genetic predisposition to some of the diabetes complications that we see. And you guys have found that nephrin 1 might be one of those genes. Are there any other key findings or implications from this work that you'd like to talk about? Jeff, I'll start with you.
I did want to mention Chris O'Connor from Marcus Bitzer's lab. He did a lot of the hands-on work for the morphometric analyses of these many, many kidneys. Thank you to him. I wanted to build on what you were just saying, that so in other studies and our work in the Kidney Precision Medicine Project, it's very obvious that diabetic nephropathy is quite heterogeneous. You don't always get expansion of the mesangium and enlargement of the glomerulus. You can see more vascular damage or more tubular interstitial in patients. And of course, we know that some patients that have type 2 diabetes, they don't necessarily get diabetic nephropathy. Why is that? What are the reasons? And that, finding a gene candidate, or in my opinion, I think a cell candidate, like what's going on in the podocyte, can we research into that more and figure out, can we tell when one patient may progress versus one that won't before they actually do progress? Because it's very difficult. And a lot of the things that we're seeing is, kidney biopsies are not done in patients with diabetes. They're just watched for a while. And often by the time we get a kidney biopsy in those patients, it's pretty advanced. And that the reason for that is that it's thought that it's more risk than benefit to actually do the biopsy. But there's so many things going on that studies like this could suggest that maybe we should be getting these biopsies earlier to better understand, what's going on and what are the structural features that are changing, to integrate that with the molecular features that we can get from the tissue.
Yeah, that's very interesting. Alan, do you have any favorite key findings?
Well, what happened one time, I was visiting Ann Arbor, actually, going out to dinner with Scott Suleimanpour, who's a very outstanding diabetes researcher at the University of Michigan. And he just casually said to me something that I didn't know, which is that diabetic nephropathy is more common in type 2 diabetics than it is in type 1 diabetics. That really stuck with me. And I asked him, well, why? What's going on there? And he speculated that maybe it's because type 2 diabetics have dyslipidemia and type 1 diabetics usually don't. Almost everybody who has type 2 diabetes has hypertriglyceridemia.
Yeah.
And the BTBR mice have pretty severe hypertriglyceridemia, and the B6 mice do not. And it's interesting, by the way, because it's inversely correlated with liver triglyceride. So the B6 OB mouse has a fatty liver, and the BTBR mice do not. And in fact, we published a paper years ago speculating that gluconeogenesis and lipogenesis might be in competition, and that one occurs at the expense of the other in these two mouse strains. So we thought, well, why don't we do an intervention experiment? Why don't we see if we ameliorate the hypertriglyceridemia in the BTBR mouse, will we ameliorate the nephropathy? So we got an antisense oligonucleotide from Ionis to ApoC3, which is an inhibitor of lipoprotein lipase, and has been used clinically in humans. It's out there to alleviate hypertriglyceridemia. And we injected it into the BTBR mice. We alleviated their hypertriglyceridemia, but we did not alleviate the nephropathy. So we got a negative result. It was disappointing, but we did publish it. I think it's an important negative result.
We've discussed your favorite key findings and touched a little bit on future directions even. Do either of you have a final take-home message that you'd like to impart here? And I guess I'll start with Jeff.
Well, I mean, I think my take-home message, we have already touched on that, there are genetic features that if we could discover to figure out which patients are more likely to progress, that would be a huge step forward. And this study suggests, but as Alan pointed out, doesn't prove that the podocyte and injury to the podocyte might be crucial to better understand in diabetic nephropathy in order to do that.
Alan, anything else?
Oh, I think it's a shout out for mouse genetics.
Sure.
And for the granular phenotyping we can do in animal models that is just impossible in humans, and the obviously much greater control over environment that we have when we work with animal models. We're doing a very big diet study right now that is enabling us to do things that just would be impossible in humans.
All right. Thank you so much, Doctors Hodgin and Attie, for sharing your expertise with us today and discussing your recent work highlighting the identification of nephrin 1 as a gene that drives susceptibility to diabetic nephropathy. I encourage the listeners to read the full article in the February issue of Diabetes. Thank you.
Thank you.
Thank you.
Welcome to this edition of Sweet Talk, where we're exploring the work of Dr. Frans Schuit and its implications for our understanding of diabetes. Dr. Schuit's research from 1998 contributed to rethinking the role of glucagon, traditionally viewed as a counterbalance to insulin. His work suggested that there's a more nuanced relationship. Joining us today are the authors of the Classics in Diabetes article, Dr. Jonathan Campbell and Dr. Frans Schuit. Welcome to both of you, and thank you for being here today.
Thank you.
It's a pleasure.
And Dr. Schuit, your study laid a foundation for rethinking a traditional role for glucagon. Maybe you can briefly take us through this evolution, maybe a historical context leading up to the studies, and then maybe what led you to the hypothesis and the ultimate conclusion.
Okay, thank you. Yeah, I think the biggest root of this work, of this tree, is the complexity of the pancreas, the endocrine pancreas. As you know, when you start to think about the basic units, the pancreatic islet, it's really a complicated microorgan. There are several endocrine cell types with their specific hormones. That is, of course, one type of complexity. But then the circulation of the islet is really amazing. When you calculate this circulation as the amount of blood per gram of tissue per time unit, this is a level of circulation that exceeds almost anything you can compare with. So there's a high blood flow in this system. And then lastly, there is a really big, intense level of innervation by the parasympathetic and sympathetic nerve system that also have a very powerful influence on the function of the islets. And then there are many things that we really don't understand in this complexity. What is the local environment of the cells, the interstitium? How are the cells, collaborating in subunits? The heterogeneity of beta cells. So there are very many difficult problems with a complicated biological system. And finally, of course, there's not one islet. There are thousands of islets in rodents and many more in humans. So this is a challenge when you want to do biochemistry or molecular biology. It's really a difficult problem. So this was certainly a basis for the environment when I started in the early 80s, my PhD in the Diabetes Research Center at the Vrije Universiteit Brussels in Belgium. And my mentor had thought about this already quite a bit, Danny Piepeleers, and he developed techniques to go to a much more simplified system, which is the purified red beta cell system. And also we had non-beta cells to compare with. And they were produced in sufficient quantities, let's say millions of cells from collagenase islets, pancreatic islets, so isolated islets. That was a system that was already available for more than a decade by then. So this, of course, really diminished the complexity to an extreme end, a purified cell system. And it was really surprising when insulin secretion was measured in that system, that glucose was a poor secretagogue. There was little insulin release when stimulated with glucose. And this was not due to poor viability or poor integrity of the cells. The morphology was fine. They could be cultured. The viability remained high after culture. But that was not the problem. An attempt to, a shot in the direction was, could it be the other endocrine cell types? And so Danny did an experiment where he co-stimulated the beta cells with glucose and glucagon. And that really restored the level of insulin secretion to what was expected from the isolated islet or the intact pancreas. And that was actually the basis when I entered the lab. That was the understanding of what was going on. So I studied cyclic AMP at that moment. That was 1985. We published about that. And like insulin secretion, cyclic AMP was really low in the beta cells compared to islets. And when you added glucagon in nanomolar concentrations, you came to the levels seen in the intact islet. So it was as if glucagon was lacking in the isolated beta cell system, while it was, of course, present in the isolated islet and maybe also in the intact pancreas. And so a colleague of mine, Frans Schuit, he did radioligand binding studies, hormone receptor binding studies, and he really could demonstrate the presence of glucagon receptors on beta cells, on the pure beta cells. So that was, of course, all done long before the GLP-1 field evolved. There was no mention of GLP-1 or GLP-1 receptors on beta cells. And then a few years before we started the classic study, in 1993, Kofod et al. Published using a beta TC3, a mouse beta cell line, that glucagon was actually stimulating insulin release via not glucagon receptors, but GLP-1 receptors. And that made us think what's going on. But this was a little bit controversial because other studies really said the opposite, that glucagon stimulated insulin release via glucagon receptors. So this was an interesting topic of controversy. And when we look back to the data that Frans Schuit did, actually what you could see in the radioligand binding studies, when you convert binding to a scattered plot, that the scattered plot was not a linear line, which can give you then the affinity and the number of sites per cell, like in the liver system, but that it was a curve. And the curve could be fitted with two lines, one line representing a population of high affinity receptors and another population representing low affinity receptors. So two receptors for the same hormone. That was the basis actually for our thinking. And then, of course, we started to analyze this. There is a diabetes paper we published in, I think, 1996, also authored by Karen Moons et al., where we used the new techniques of molecular biology, northern blots, etc., western blots, to demonstrate that both glucagon receptors and GLP-1 receptors are expressed and functional on these pure red beta cells. So that was the beginning. And then in the classic paper, what we did, we combined radioligand binding studies again, using iodine-labeled glucagon and GLP-1, and also specific receptor antagonists, so peptide analogs that can block competitively with their hormones on their own receptor. So there's a glucagon receptor analog, I think was a deshistidine-1 glucagon analog, and then, of course, the GLP-1 receptor antagonist, the exendin 39A mite, which only interferes with GLP-1 receptors. And in that, working that way, we could really show that glucagon binds with high affinity to the glucagon receptor and with low affinity to the GLP-1 receptor. And then we did, of course, a cyclic AMP analysis. And yeah, we saw that both receptors can contribute to the second messenger production in the cell system. And so the interpretation, I think it was straightforward, but it is still a matter of discussion that the high affinity receptor is capable or competent to detect circulating glucagon, while the low affinity receptor, the GLP-1 receptor, may be adapted to detect interstitial glucagon concentrations, which probably are much higher than the circulating. And this is interesting because for insulin, Chris van Schravenegg has shown before that there are both IGF-1 receptors and insulin receptors in the islets. And the IGF-1 receptor is actually a low affinity insulin receptor, so you may have the same there. And for somatostatin, I did some studies for somatostatin. You also have different somatostatin subtypes in the islets, and one has a high affinity and the other a low. So the glucagon system may not be the only system that is working that way. And that was where we ended in 1998. So this is a brief summary of the roots, I think.
Perfect. Yeah, that was a really great foundation and understanding of what was going on at the time. I want to fast forward a bit for Dr. Campbell, and I was wondering if you could reflect on Dr. Schuit's early work and how this helped or currently helps to guide your current research interests. I believe you and Dr. David D'Alessio have an article coming out in the March issue of Diabetes that has a direct relationship to this topic. Maybe you can give us a sneak peek on that work.
Yeah, we can certainly walk through where it was left off and to where we are today, including the article that you just mentioned that's about to come out. We got into this game sort of trying to understand why there's a glucagon receptor on beta cells. And we used more modern techniques. So we had advanced past northern blots a little bit into mouse genetics and ended up knocking out the glucagon receptor from the beta cells, trying to figure out what that does. And it turns out that mouse model had no phenotype, as if you lose the glucagon receptor, there's no effect on beta cells, no effect on glucose tolerance. And when you get a null phenotype, what do? You go back into the literature and try and figure out how to do it. And this is where we stumble across all of this old literature, that we're not discovering, we're rediscovering these concepts that glucagon has more than one receptor on the beta cell where it can drive insulin secretion. And our world changed when we started knocking out or blocking the GLP-1 receptor and having huge effects on intracellular cyclic AMP, ultimately insulin secretion and glucose tolerance, basically repeating a lot of what was just discussed. So we kind of stumbled across this and rediscovered this important axis. And since then, we have started to march forward asking questions about why would the alpha cell stimulate insulin secretion? And I don't know that we have all the answers to those questions at this point. We've made some significant progress showing that the alpha cells are a target for gut incretins. So the incretin that people don't think a lot about is GIP, where they're probably thinking a lot more about it now with tirzepatide. But up until GIP has reemerged as an important incretin, it was always GLP-1. And well, it turns out that GIP talks to alpha cells and kickstarts this alpha to beta cell communication through the GLP-1 receptor. And it can talk to the beta cell or the alpha cell to stimulate insulin secretion. So in both halves of those are really important for postprandial metabolism. So it looks like glucagon secretion is a key contributor in postprandial metabolism for insulin secretion. The paper that you are referencing is all of the work that we and others had done up to this point was really looking at this in the context of rodents or isolated islets, and we wanted to translate this into humans. And there's a lot of work that says that glucagon can stimulate insulin secretion in humans. We just kind of wanted to try and understand when and how and what the implications of that access might be. So we took healthy humans and we infused pretty high doses of glucagon. It's tricky when you do those studies because the anatomy and the geography of the systems are backwards. So normal physiology is the alpha cells are juxtaposed right next to the beta cells, and that's how the system works. So when they secrete glucagon, the first thing they do is they see a beta cell, and then it leaves the islets and the pancreas and travels to places like the liver, where glucagon has its most well-known actions to regulate hepatic metabolism. When you do the kind of opposite study where you infuse it into a vein, which is what we're doing when we're giving glucagon to healthy humans, it's going to see the liver well before it sees the islets. Moreover, it's going to be diluted by the time you get to the islets. So we had to give quite high concentrations of glucagon to try and mimic what we would call intra-islet concentrations of glucagon. And if you do that, it robustly stimulates insulin secretion in humans that we can measure. In fact, one of the things that I was taken back of when the data started coming out is we would take people and they'd either be fasted and we give them glucagon, or we would raise their blood glucose up to a postprandial level. So you get glucose-stimulated insulin secretion, and then we give them glucagon. And the magnitude of insulin secretion that occurs when you give glucagon at high glucose far outweighs the effects of glucose alone. So just like Frans was saying, is that glucose is not a great insulin secretagogue. It is a necessary insulin secretagogue. It's certainly a critical component of the response. But alone, glucose-stimulated insulin secretion is pretty poor. And that's what you would see if you took isolated beta cells and tried to study their insulin response to glucose alone, absent something that's going to drive cyclic AMP like glucagon. And as soon as we give glucagon back, the amount of insulin secretion that you see is vastly superior. And so that was one of the surprising findings from this paper. But what we also saw, just to sort of fast forward and try and understand and contextualize why glucagon might be an important component of postprandial metabolism in this paper, is we found that we saw glucagon was doing two other important things with respect to insulin. One, it was enhancing insulin sensitivity in the liver. So we see this, and Kirk Haberger has made some really important insights here showing that glucagon and insulin are not opposing factors in hepatocytes, but they actually might be coordinated, and glucagon can drive insulin sensitivity. So we picked that up in this paper in our human studies as well. And then one other thing that we saw that we have a little bit of data to at least shine a light on is that it might be that glucagon is decreasing insulin clearance. It's allowing insulin to get past the liver. So as you may know, first pass insulin clearance by hepatocytes can be up to 50%. And that seems to be a regulated system that sort of gates how much insulin is making it to tissues beyond the portal circulation. And our results from this studies in humans say that glucagon might be enhancing the amount of insulin that makes it past the liver. And the interpretation is that more of that insulin is going to regulate signaling in peripheral tissues like muscle or adipose tissue or whatever your favorite cell type is. So with that in mind, it seems like the rise in glucagon that you would see in a postprandial state may be more than just stimulating insulin secretion, but also dictating what that insulin is doing at the liver and beyond.
And following up on this, Dr. Schuit touched on this a bit, but I think there is the fascinating puzzle here in the system related to glucagon and GLP-1 signaling that many might think about. And I'd love to get your insight on this. Essentially, both GLP-1 and glucagon may act via the GLP-1 receptor with a low affinity, maybe more of a lower affinity of glucagon at this receptor, and this might be unidirectional. So the question is, how does GLP-1, which is a gut hormone that's secreted at relatively low levels, and then glucagon being secreted during fasting at higher levels, both end up acting on that same receptor? And then what do you think is happening behind the scenes of that interaction? What could be the underlying wisdom of such a system?
It's a very difficult question, and I think one of the unknowns, and I think it's debated whether or not a lot of gut-derived GLP-1 actually can reach the beta cell. This is debated at this moment, and I think that's a good point. The other point that is debated actually is whether, and we have skipped this part in the story so far, actually the gene responsible for glucagon production is actually also responsible for GLP-1 production. This is a pre-pro hormone system which encodes several peptides, then depends on the proteolytic enzymes which are present inside the specific cell type, which peptide actually is the final outcome. Of course, this is not the only system. The pro-opiomelanocortin system was the first that was discovered in this way. But the pre-pro-glucagon gene actually encodes not only glucagon, but also GLP-1 and GLP-2. Now, of course, in textbooks explanations, the distinction between the various proteolytic processing enzymes is black or white. It's all or nothing. But in reality, probably nothing is perfect, and there may be little bits of GLP-1 produced by alpha cells. And actually, this has been demonstrated by some groups or claimed to be important by some groups. So this is also a matter of discussion, whether the local environment that starts with the alpha cell actually may end up with little bits of GLP-1 reaching the beta cell. That is a possibility, of course. And it could well be that depending on the state of the organism, the amount, even if it's a little bit, that the amount of GLP-1 that is locally produced may vary from time to time or for situation to situation. So that is one thing I think that needs further investigation. Another important variable, I think, is the discussion about the fact whether or not the circulation, the microcirculation in the islet is well suited to allow a flow of information from alpha cells to beta cells. And actually, I'm sure that you are aware of the perspective in the December issue of Diabetes, authored by Susan Bonner Ware and Gordon Ware, where they actually make a point in stating that the anatomy, the static anatomy, if you look at the microcirculation, that the arterioles actually first reach the beta cell core of the islets, so that the blood flow goes from the beta cell to the alpha cell, not in the opposite direction. So that is a challenge, of course, if you want to understand what is exactly paracrine glucagon reaching the beta cell. I remember, if I may speculate about this problem, which is a really difficult problem to understand, I remember studies of in situ circulation, where you can actually see through a microscope the surface of an islet on the surface of the pancreas. And I remember images where in certain vessels, microvessels, the circulation direction reverses from one moment to another. So that may add to the complexity. So it's a difficult problem. And thirdly, there is, of course, this idea that there are microdomains in the islets where cells are electrically coupled by gap junctions, so that it is sufficient for one beta cell that is close to an alpha cell to be stimulated by glucagon or GLP-1, and that via electrical coupling, it can trigger other beta cells to collaborate in the same insulin secretory response. So these are all, difficult issues and complex issues to further study.
I think Frans brought up a couple of really good points, some open questions that need to be addressed. One is there is a two receptor problem. Why would a system develop two receptors that look very, very similar in terms of their function to receive the same message? So why would glucagon signal through two receptors? Are they truly different? I don't know. And I think that's an important question to answer. The other point that Frans brought up is there's the potential for two hormones. Why would the alpha cell make two hormones? Because there is pretty strong evidence that alpha cells do make GLP-1. Is it biologically relevant amounts of GLP-1 is an open question that I think is being pursued as we speak right now. But it does beg the question of when would an alpha cell want to secrete glucagon and when would an alpha cell want to secrete GLP-1? In our hands, we see that the levels of glucagon are about 300 times higher than the levels of GLP-1 in a rodent islet. On the other hand, GLP-1 is about 300 times more potent than glucagon at stimulating insulin secretion. So you could make an argument, depending on the data you want to use, that either could be a means of communicating from the alpha cell to the beta cell. The situation that Frans was alluding to is one answer to that question is this plasticity in the alpha cell to switch between hormones may be a mechanism for it to compensate based on the environment. If you can tune up alpha to beta cell communication by increasing the production of GLP-1, maybe by changing the levels of the pro-hormone convertases, it may enhance insulin secretion to overcome peripheral insulin resistance. So this may be a compensatory mechanism that the alpha cell engages in order to increase beta cell function. That's an option. I think it's a hypothesis that does need to be tested. But it does tell you that having the alpha cells have this plasticity is probably very important for beta cell function. Where it sits in the spectrum of metabolic disease has yet to be seen.
Perfect. And as we're wrapping up, I'd like to ask both of you, what do you view the next big question or frontier in glucagon research? I know Dr. Campbell mentioned the incretin-based therapies currently being developed, currently on the market for diabetes and obesity treatment. What might be the next significant breakthrough in diabetes treatment considering the current trajectory of research?
Well, I can start on that because I think if you look at the last 50 years of glucagon biology, in the 70s, they were a lot of effort was being put into developing glucagon receptor antagonists to target hyperglycemia. And that was pursued up until probably about 10 years ago. And a lot of that effort progressed quite deep into clinical trials that were really good at lowering glucose, but came with a lot of other undesirable effects on lipid metabolism and alpha cell hyperplasia that just stalled the development and basically killed it. And what we've seen in the last 10 years is a 180-degree flip-flop where now, instead of developing glucagon receptor antagonists, glucagon agonism is being pursued. It is being paired with incretins with the concept that you need the insulin function of an incretin to counterbalance the hyperglycemic actions of glucagon. But in doing so, you can leverage glucagon's biology to drive energy expenditure, lipid metabolism, probably a lot of other healthy benefits that it has for the liver as well, and get either positive benefits on weight loss or liver health. And so the shift has gone from blocking glucagon to enhancing glucagon, which sets the framework for we don't really know anything about anything. Because if you do a 180-degree swap, you probably don't understand the system well enough. So just trying to get a better feel for what the biological role of glucagon is going to inform on the therapeutic potential of that system.
If I may add perhaps one idea, we have for diabetes research mostly focused on rodent models and human studies. This is really a bias in the way data have been produced over the past decades. If you look at glucagon, it is an evolutionarily very old hormone. So it's practically, I think, in all vertebrate clades, it is present. And perhaps a better idea of the evolutionary role of glucagon function may give us more insight of what's going on. What has been the first function of glucagon? Has it been to counteract insulin, work on the liver? So I think that is really interesting. And why I think about it is that, for instance, in birds, there is a very different organization of the endocrine pancreas. And there is actually a glucagon-rich pancreatic lobe, which is almost entirely composed of glucagon cells. So there are vertebrates with a very different endocrine organization. And I think studies aim to understand this better may learn us new things where we haven't thought of. This is just an idea. And I mention that because the last years I was working in Leuven, we did a comparative analysis of vertebrate genomes, studying quite a large number of genomes, and we stumbled upon very interesting clade-specific differences between the lineages. So reptiles, including birds, mammals, they are really different in some ways in the architecture of the genome. And this I think may add something to understanding glucagon and glucagon function. But it's intuitive. I don't know exactly why it would be, what it would result with, but I think it is a possibility that may learn us something new.
I think that's a really important point. So the textbooks say that glucagon is a counter-regulatory hormone to prevent hypoglycemia. But hypoglycemia is practically a century-old problem. It really wasn't an issue until insulin was developed, and then insulin overdosing drove hypoglycemia down. Unless you were a caveman that stumbled across a honey patch, it was very unlikely that you were going to have hypoglycemia, which means that hypoglycemia and glucagon are probably not linked as an evolutionary pressure. So what is the evolutionary pressure to develop glucagon biology? Is it insulin secretion? Maybe. Is it completely independent of glucose metabolism? Probably. And what that is, I think is part of the future. So if you want to get away from what is the pharmacological potential of glucagon and you just want to understand basic biology, I think that is an important question to ask. What was glucagon doing in our ancestors that was an evolutionary pressure that was absolutely essential, because it probably was not glucose metabolism.
Yeah, that's an interesting point. To go on with birds, what really amazed us is that basal glycemia in birds could be defined as highly diabetic when you would use the human standard. So their basic glucose level is 10 millimolar or higher. I think the expansion of animal models to other systems than the rodents and humans has a high chance of learning us something new. It's my gut feeling.
Fantastic. Thank you both, Dr. Campbell and Dr. Schuit, for joining us today. I really enjoyed this discussion. To our listeners, thank you for tuning in to this edition of Sweet Talk. Dr. Schuit's original research article titled Dual Glucagon Recognition by Pancreatic Beta Cells via Glucagon and Glucagon-Like Peptide 1 Receptors can be found in the January 1998 edition of the journal. The Classics in Diabetes article highlighting this work, titled GPCR Promiscuity, Reshapes Islet Physiology by both Dr. Campbell and Dr. Schuit, can be found in the September 2023 edition of the journal. And finally, the paper of the month from the groups of Drs. John Campbell and Dave D'Alessio, titled High Doses of Exogenous Glucagon Stimulate Insulin Secretion and Reduce Insulin Clearance in Healthy Humans, can be found in the March 2024 edition of the journal.
Thanks to you all for joining us on our second Diabetes Bio podcast. We hope that you enjoyed this little snippet of all things from the Diabetes Journal and what it has to offer this month. The feature article that we discuss each month is freely accessible at diabetesjournal.org/diabetes. And this is also listed in the episode notes for your reference. We look forward to presenting to you next month.